whenever you say biomolecules, these are the polymers. These are the macromolecules or the big molecules in our body that support life. Okay, hence we use the term biomolecules. Now, we said there are 4 types for this. You have that on your slide. You have your carbohydrates, your lipids, your proteins, and your nucleic acids. Now, as part of your warm-up or your review today, you were asked what the differences are and what the similarities are between your carbohydrates and your lipids. Okay, so one similarity that you can find here, of course, is in their structure. As you can see, your carbohydrates and lipids, both of them are made up of carbon, hydrogen, and oxygen. Okay, so both of them have carbon, hydrogen, and oxygen. The difference is that in their proportion. Okay, now remember we said in your carbohydrates, the proportion of carbon, hydrogen, and oxygen here is in 1:2:1 ratio. Okay, but in your lipids, there's going to be more atoms of hydrogen as you compare it with your carbon atoms and also with your oxygen atoms. Okay, so that is one difference between your carbohydrates and lipids. Another difference, of course, is that your carbohydrates are water-loving molecules. They are hydrophilic, while your lipids are hydrophobic molecules, okay, which means that they don't like to be in contact with water. Okay, now going to the different monomers that we have here. Again, when you say mono, this means single unit. So when you say monomer, these are the single structures that you can find in all these biomolecules that you have. Now, again, the monomer of your carbohydrate is a monosaccharide. Okay, it's monosaccharide or simple sugar. Now, as for the uses, your carbohydrates, we said this is the quick source of energy, okay? This is the fast source of energy in your body. Whenever you need energy, your body is going to be going to use up your carbohydrates, okay? Now, again, another important use of your carbohydrate is that it is also as storage. Okay, so example for this would be your starch in plants and glycogen in animals. Okay, so these are both storage forms of carbohydrate. Another thing is that they also form structures. Okay, so example would be your cellulose. This is a structure that you find in your plant cell wall, and chitin, which is the structure that you find in the exoskeleton of insects. Okay, so again, these are the different uses of your carbohydrate. Monomer would be your monosaccharide or simple sugar. Now for your lipids, your monomer here would be 1 molecule of glycerol. Okay, so 1 molecule of glycerol and 2 fatty acids. Okay, so those are the monomers of your lipids. As for the uses, again, just like your carbohydrate, this is also a source of energy, but this is a more concentrated form of energy and it is stored. Okay, which means that again, if your body needs energy, the first thing that it is going to consume would be your carbohydrates. If your body has no carbohydrates anymore, then that's the time that it is going to target your lipids, because lipids again are just stored energy, more concentrated form of energy. Another function is that it keeps us insulated. That means it keeps us warm. It also forms structures. Okay, so your phospholipids in your cell wall are made up of your lipids. You also have your sex hormones, your steroids are examples of lipids. Okay, so we are going to take a look at the different types of lipids later. Now, as for your proteins, you have your carbon, hydrogen, and oxygen still, but most importantly, it has the nitrogen atom. Okay, it contains nitrogen atoms, and in some cases, it might have sulfur. It might even also have phosphorus. Okay. Now for the monomer of your proteins, there are amino acids. Okay. So monomer would be your amino acid. For the uses of your proteins, first one, it's very important for the cell production, making of your muscles. Okay. So that's also your proteins that would play a very important role there. Another thing would be for structural forms in your body. You have your actin and your myosin. These are structural proteins that you can find in your muscles. You also have keratin in your hair. Your fingernails also would have keratin. Now, another thing that is very important under your proteins would be your enzymes. Okay, so all enzymes are types of proteins, and of course, their function is to speed up chemical reactions. Okay, so proteins would also include your enzymes. Now, the last type of your biomolecule here would be your nucleic acids. Now, aside from carbon, hydrogen, oxygen, nitrogen, this also would contain your phosphate group or your phosphorus. Okay. Now, for the monomer of your nucleic acid, you have your nucleotides, which is composed of three different parts. Okay, the first part would be a ribose sugar. This is a type of sugar with 5 carbon atoms. Okay, it also has a phosphate group and also has an N basis or nitrogen basis. Now you have two types of nucleic acids and they play very important roles in our body. If it is an RNA, your ribonucleic acid, its function is for the making of proteins. If it is your DNA or deoxyribonucleic acid, its function is for heredity. Okay, so it passes on the genes from the parents to the offspring or from the parents to the kids. Okay, so again, this is a summary of all the biomolecules. We've already talked about this yesterday. And today we are going to be talking about the different types of each of these biomolecule. We are also going to look at their structure. Okay, sometimes your EOC test is going to show you a certain structure, a formula or a chemical structure, and it is going to ask you what type of molecule you are looking at. Okay, so it's very important that we know the structures of these types of biomolecules. All right, so we start with the first one here, which is carbohydrate. Okay, so again, when you say carbohydrate, you have your carbon, hydrogen, and oxygen. We said the monomer for this, the simple unit, the simplest unit of your carbohydrate would be your monosaccharide, okay? So you have your monosaccharide, that's your simple sugar. If you have two monosaccharides combined, you'd call that disaccharide, okay? So di here would mean two, okay? Sometimes you may also see the term oligosaccharide. Oligo here means few. Okay, so when you say oligosaccharide, it is a form of sugar, form of carbohydrates that has a few units of sugar. And you also have number 3 here, polysaccharide, that means many units of sugar together. Okay, so these are the 3 main types of carbohydrates. Monosaccharide, only 1 unit of sugar. Disaccharide, you have 2 units. So that's monosaccharide and monosaccharide combined. And you have your polysaccharide. These are the bigger molecules of carbohydrates that you have. Now, the specific examples of your monosaccharide can be found on the next column here. For your monosaccharide, the first example will be your glucose. Okay, so glucose is the same thing that is produced by your plants during photosynthesis. This is also called, in humans, this is called blood sugar. Okay, so this is the type of sugar that is measured whenever one of your family members has diabetes. Okay, if your family member has diabetes, what is being measured by that machine that they use to measure blood sugar is going to be glucose. Okay, that's blood sugar. Now, another thing that you have here would be your galactose. Okay, galactose here is known as brain sugar. So this is the sugar that is used up by our brain for it to function properly. Okay, so that's your brain sugar or galactose. You also have the third type, which is fructose. Okay, so fructose here is also known as fruit sugar. Okay, this is a type of sugar that you find in fruits, and this is the sweetest form of sugar. It is naturally occurring. That means it is not made in the lab. You find this in nature, specifically in the fruits that we eat. Okay, so that's your fructose. Now, another type of monosaccharide is ribose. This is a 5-carbon sugar, and you find this, of course, as very important part of your nucleic acid. Okay, so we are going to also discuss that later. So again, these are the 4 different types of your monosaccharide. Again, monosaccharide, there's only 1 unit of sugar that you have. Okay, now we go to your disaccharide or oligosaccharide. When you say disaccharide, again, you have 2 units of sugar. So this is a combination of your different monosaccharides here. For the first one, maltose. Maltose is also called malt sugar. This is a type of sugar that you get from your energy drink. This is the type of sugar that you get from your cereals. This is also the type of sugar that you get from the grown-ups would get from beer. Okay, this is also sometimes called beer sugar. Okay, that's maltose. Now, we said that this is a disaccharide. That means it is a combination of 2 monosaccharide. For your maltose, this is a combination of 2 glucose molecules. Okay, so if you have glucose and glucose combined, you form a disaccharide, and that type of disaccharide would be maltose. Okay, so that's for your maltose. Now, another form of disaccharide would be your sucrose. Sucrose here is called your table sugar. Okay, so this is a type of sugar that you use when you're cooking. This is a type of sugar that you see in your kitchen. Okay, so that's your sucrose, your table sugar. This is a combination of your glucose plus fructose. Okay, so glucose plus fructose, that is going to give you sucrose, a disaccharide. Now, the last type of disaccharide that we have here would be your lactose. This is also called milk sugar, the type of sugar that we get from milk. And this is a combination of glucose and galactose. Okay, hence the term lactose. So again, these three are the different types of disaccharide that you have. Okay. Now, as for your polysaccharide, again, when you say polysaccharide, these are bigger molecules of carbohydrates, a long chain of your monosaccharide. And you have four types here, cellulose and chitin. Of course, you know this to be your structural carbohydrates, cellulose in plant cell wall, and chitin in the exoskeleton of your arthropods, of your insects. Now, your starch and glycogen are two examples of storage carbohydrates. These are stored carbohydrates. Starch is found in your plants, and of course, glycogen is found in your animal cells. Okay, so these are the different types of carbohydrates. So we have three very different types: monosaccharide, only one sugar; disaccharide, combination of one sugar. So you combine one sugar and one sugar to make a two chain or a two unit of sugar. Then of course you have your polysaccharide, longer chains of carbohydrates, longer chains of sugar that you have. This next slide is showing us the different forms and different structures of your carbohydrates, okay? It looks very complicated, but let me explain it to you. Now, the first two that we have here are your examples of monosaccharide. Okay, so this is glucose here, and this is fructose. Now, as you can see, your sugars would usually have 6 sides. Okay, they usually have the hexagonal shape. Okay, so if you have one unit of sugar here, you know that this is just a monosaccharide. Okay, so that's a monosaccharide. This is just glucose. This is just fructose. Both of them are monosaccharide. Now, the second example that we have here at the bottom, you have sucrose. Now, again, we know that sucrose is a disaccharide, and you can actually that in its structure. Okay, so you have one unit of sugar here. You also have another unit of sugar here. And as you can see, they are connected, forming your disaccharide of sucrose. We go to the next type of biomolecule that we have, your lipids. Again, we said your lipids would also have carbon, would also have hydrogen, would also have oxygen. And of course, the monomer of your lipids, the simple units of your lipids would be 1 glycerol and 2 fatty acids. Okay, so 1 glycerol molecule and 2 fatty acids. Now, you have 3 different types of lipids here. Okay. First one is your triglycerides. This is commonly known as fats. Your second type would be your phospholipids, and the third type would be your steroids. Now, for your triglycerides, the term tri here, of course, would mean 3. Okay, remember the monomer of all your lipids would be 1 glycerol and 2 fatty acids. Now, in the case of your triglycerides, instead of having just 2 fatty acids, it actually has 3. Okay, hence it is called triglycerides. Triglycerides, 1 glycerol and 3 fatty acids. Now, examples for this would be your saturated fats and your unsaturated fats. We will get to this in the next slides. Now, another type of lipids that you have here would be your phospholipids. From the name, of course, it suggests that this is a combination of phosphate groups and lipids. Okay, so this is a combination of your phosphate group and lipids. So as you can see here, you have 1 glycerol and 2 fatty acid. That's the monomer of your lipid. Your lipids would always have 1 glycerol and 2 fatty acids. And added to that, it has a phosphate group. And so you call this your phospholipids. Okay, that's your phospholipids, a lipid, and a phosphate group. Now, this here, of course, is the structure that you find in your cell in your cell membrane. Okay, so plasma membrane, cell membrane would be phospholipids, and of course, your steroids here. This is a more complex form of lipids. We are going to the different structures in this after this slide. Okay, now we go to the different structures that we have. Again, this is your triglyceride. This is your glycerol component here. This one here would be your glycerol, C3O3, and your H5, okay? So this is your glycerol molecule. And again, in your lipids, you should only have 1 glycerol molecule and 2 fatty acids. But in the case of your triglyceride, instead of just having 2 fatty acids, it has 3 fatty acids. Now, for your phospholipid, again, we mentioned that this is a combination of your lipid. So your lipid component here would be this. Okay, so this is your lipid component. And of course, you have your two fatty acids, that's glycerol, and two fatty acids giving us your lipid. And it is combined with your phosphate group of your PO4. Okay, so one atom of phosphorus and 4 atoms of oxygen. Okay, so that's your phospholipid, a component of all our cell membranes or plasma membranes. Now for your steroids are more complex forms of lipids. Now it's very important for you to note the structure of steroid. The steroid always have the characteristic structure of 4 rings with your fatty acid chain here. Okay, so you'll see 4 rings and your fatty acid chain here. We go to your saturated versus unsaturated fats. We said these are both examples of your triglycerides. Now, what is the difference between your saturated and your saturated fats and your unsaturated fats? Your EOC test might also ask you this. The only difference is that in your unsaturated fats, in your unsaturated fatty acid, there is one double bond. Okay, so there's one double bond there. In your saturated fatty acid, there is none. Okay, I'm talking about the carbon and hydrogen combination, not including this one here. Okay, not including your carboxyl group. We're looking at the chain of your carbon and hydrogen, your hydrocarbons. And for your unsaturated fatty acid, again, there is a double bond. Okay, there's one double bond, which means that if you try to compare the number of hydrogen between your saturated fatty acid and your unsaturated fatty acid, your saturated fatty acid would have more hydrogen atoms. Okay, it is going to have more hydrogen atoms because when you form your double bond here, those hydrogen atoms that are actually here are lost. Okay, so those hydrogen atoms here are lost since you have a double bond. So comparing the number of hydrogen atoms that they have, your saturated fatty acids would have more hydrogen atoms. Okay, so that's why we call this saturated. This actually means it is saturated with hydrogen atoms. Now, if you try to compare these two, your saturated fatty acid is more harmful to your health. Okay, it is more harmful to your health. Examples of your saturated fatty acids are your animal fats. Okay, so animal fats like your margarine, your lard, your butter. These are examples of your saturated fats. Okay, so they're more harmful to your health than your unsaturated fatty acids. Unsaturated fatty acids, examples for this would be your vegetable oil. That's also an example of your fats, your triglycerides, but of course it is unsaturated, which makes it healthier for your body. Okay, so vegetable oil, even fish oil is part of your unsaturated fats. Okay, but saturated fats, you have your animal fats and they are more harmful to your health. Okay, so again, major difference between saturated and unsaturated fats. Unsaturated fats would have one double bond. Going to your next slide here, your proteins. There's not much that we are going to discuss about proteins today. Remember, we said your monomer for your proteins would be your amino acid, and all your amino acids would have this structure here, your carbon as a central atom, one hydrogen atom attached to that central carbon atom, and you have your amino group here, which is NH2. You also have your carboxyl group, which is COOH, and there is an R group here. Now, as I've mentioned yesterday, it is only the R group that changes from one different type of amino acid to another. Okay, so this is only this part that changes. The rest of the parts there would be the same. Okay, now as we've mentioned yesterday, glycine is the simplest form of amino acid, and for your glycine, it only has one hydrogen atom here as its R. Okay, so again, the only part that changes from your amino acid from one form to another would be this R part here. The rest of the parts are all the same. Okay, you have your DNA again in your RNA, deoxyribonucleic acid and ribonucleic acid. Both of them are nucleic acids. Both of them are made up of your nucleotides. You have your ribose sugar here. Okay, so you have your ribose. You also have your phosphate group, and of course you have your N bases. Now, first difference is in their structure. Your DNA is double-stranded. Okay, it has 2 strands. Your RNA, on the other hand, has 1 strand only. Okay, so it only has one strand. In the structure here, your ribbon-like structure that you find here, the ribbon-like component that you find here, this one, this is made up of your ribose sugar and your phosphate group. And the lines that you find in the middle, okay, those lines are your nitrogen bases. Okay, so again, the ribbon-like structure, that's your ribose and your phosphate group. The lines in the middle, those colorful things in the middle, those are your N bases. So again, your deoxyribonucleic acid or DNA is double-stranded. Your RNA only has one strand. Now, we said they also have a difference in the type of sugar that they have. Deoxyribose for your DNA and ribose for your RNA. Now, the main difference here would be when you say deoxy, this actually means removal. D is removal. So removal of one oxygen atom. Okay, so that means in your deoxyribose, in the sugar of your DNA, there is one missing oxygen atom. If you try to compare your deoxyribose here with your ribose, you can see that your ribose has an oxygen atom here, but in your deoxyribose, that oxygen atom is non-existent. Okay, so that is a difference between the two of them. When you say deoxyribose, it lacks one oxygen atom, and you can find it at this part here. Okay, so no oxygen here. There is an oxygen in your ribose. Okay, so that's another difference. Now, we've also talked about their differences in their N bases. Both of them have adenine and guanine and also cytosine. And both guanine and adenine are your purines. That means they are two-ringed. Okay, they have two rings. Your cytosine here is one-ringed. Okay, only has one ringed. Both DNA and RNA would have cytosine, guanine, and adenine. But only DNA would have thymine, and only RNA would have uracil.